Okay, ladies and gentlemen, great pleasure to be here. My name is Jonas Bergquist. I'm from Sweden, as you can see, Uppsala University, and a good friend of Macrell and Christian now, and we have been here and met all of you also, so that's a great pleasure. I've been asked to give a short lecture, and I promise it won't be so many details. My intention with this is just for you to get some ideas of what can be done, what we are doing in Uppsala and partly in US in pathology and in China in the precision medicine field, and also relate that to what we are actually doing this weekend, being part of the Equestrian Life. So that's why we selected a title called Molecular Features of the Equestrian Life, and I will try to connect both what's going on in human physiology with what actually can then be measured in horses and when we are in contact with horses. So this is a sort of a startup of a collaborative project that we are going to start with Christian and Macrell. So my basic background is in chemistry, biology with medical applications. So at Uppsala we do a lot of measurements, develop tools for measurements, and then we apply it to different categories of diseases in humans. And most of the things we do is in neurodegenerative diseases. So you all are familiar with Alzheimer's disease, Parkinson's disease, multiple sclerosis, and lately also a big focus on something called myalgic encephalomyelitis, which is not so well recognized yet, but it's called ME in short, and that's a disease, neuroinflammatory disease that we are working on right now to try to understand more. So please go ahead, ask questions if you have any. Just interrupt me. This is very informal, as my clothes are very informal. So I just put on. I'm normally not lecturing in riding clothes. I lectured once in a diving suit, and I was almost as sweaty as this, I have to say. Normally, well, it's hard to see. I dress up a bit, so this is how it looks when I lecture at NIH, for instance. It looks like I was going for a candidacy of the new presidentship in US. I'll make America great again, you know. But, what we are working with is the brain. So the brain is the most complex and also most fascinating organ you have to deal with. And all of you are familiar with the amount of cells that are in there, the nerve cells. There are 100 billion nerve cells that communicate with each other. And each single cell is used almost all the time because we have to keep them activated. If we don't activate the brain, it will just, as a muscle, waste. So the idea that you hear sometimes that, well, we are only using like 50% of our brain capacity, that's not true. We're actually using all of the brain, and we're using it constantly to keep it activated. But in that sense also, we need to keep the brain clean in some way. So we have pieces of time during the day where we actually are relaxing the brain a bit and rinsing the brain. So I will talk a bit about that. And there is a new system that has been more or less discovered just last couple of years called the glymphatic system, which is a waste handling system within the central nervous system. And that's something that we have been working with now a bit. However, it's not easy to measure things from the human brain in alive individuals. Not that many people want to voluntarily have someone drilling a hole in your skull and taking pieces of the brain. That's unfortunate for us, but that's how it is. So we need to look at the living brain from some different other perspectives, right? So what we have been developing in our research is ways of monitoring the brain, and we do it by something called microdialysis. It's very hard to see on the screen, I'm sorry for that. It's a small probe, so actually you have to drill a hole down to get through the bone structure, but then it's like a thin needle about 2 millimeters thick, and you put it into the tissue you want to monitor. It could be a peripheral tissue, it could be a liver, it could be fat tissue, it could be kidney, but also in the brain. So we use this in clinical routine now for patients with traumatic brain injury. Hopefully none of you guys will happen to come to neurointensive care and need that kind of treatment, but this is one way to monitor how a brain is actually coping with a situation where you have an injury. And you can then take decisions based on the molecules that we can filtrate out from the damaged tissue, and also avoid ischemic events, which is lack of oxygen, for instance, or lack of energy to the damaged piece of the brain. Another technique that we are working with is going through the eye. So, eye is one of the parts of the brain you actually can see, and see with also. So you can actually monitor things that's going on inside the brain by looking into the eye. That you can see reflections of damaged vesicles, blood vessels in the brain by looking into the eye. But we also can do monitoring of liquids that we have. So for instance, we have a technique now, so we can go in and take the liquid in the front of the eye. It's called the anterior chamber liquid or humeral liquid here. And this is a small volume, about 10 microliter volume that we can draw a sample from. You do it by a small catheter, no small needle. Or we can take sample from the vitreous body here, and then we can do monitoring of certain proteins and small molecules. And that can tell us if there is a neurodegenerative disease ongoing, if there is a higher activity of, for instance, oxidative stress, which monitors free radicals basically that are making trouble for the proteins. And then proteins, what happens with the protein when it gets modified is that it starts to behave unnatural. So it starts to clump together to each other. It binds together and form fibrils or stacked protein structures. And those protein structures are damaging the tissue, so it kills the cells around it. So can anybody come up with this kind of disease where you have this kind of phenomena? I mentioned it once already. Alzheimer's disease. I think that's the most commonly known now, that in Alzheimer's disease brain we have inclusions of proteins that all of us are carrying, but in patients with Alzheimer, suddenly those proteins stick together and then they start killing the cells around it. And an Alzheimer's brain is, if you take post-mortem analysis of an Alzheimer's brain, it's actually lost up to 60% of the weight. So a normal healthy brain in a human is about 1.5 kilograms. I can't convert it to pounds, I'm sorry for that. And in an Alzheimer's brain you have maybe 600 grams left.
Is it metabolic brain loss?
In the end, it's actually systemic, so you actually lose. I mean, in the beginning, you lose certain regions like hippocampus and so on, but otherwise, you lose all the pieces of the brain in the end, actually. So it's just wasting. We have another way of looking at the brain, and that's actually isolating small lipid structures, small vesicles that are circulating in the body, and we can pull them out from blood and then isolate them. And all organs in the body and all cells in the body, they actually excrete specific vesicles that carry signal molecules from a specific organ. So if you have a damaged organ, let's say you have a cancer, we can monitor and see these vesicles in the circulation and then do diagnosis based on these. So it's like a liquid biopsy. You don't need to go into the tissue to pull a piece of the tissue. You actually select something from circulation from blood in a simple blood test. Our favorite liquid that we are working with, my favorite liquid is this. Red wine is also okay, but cerebrospinal fluid, and that's the liquid that surrounds the central nervous system. I will talk a bit more about that specific liquid, because that's one way for us to actually monitor the chemistry in the brain without actually taking a piece of the brain and extracting molecules from the tissue. So cerebrospinal fluid you get by lumbar puncture. Has anybody had lumbar puncture at some point? Yeah. I mean, it's not pleasant, right? It's an invasive sampling technique, but it's not too bad. I mean, you get the local anesthesia and you can draw the liquid in the lower region of the back, so lumbar region. Or if you have specific interest in the ventricle cerebrospinal fluid, you have to go through the neck, and then you go up and take more central nervous system located liquor or cerebrospinal fluid here, basically. But I will get back to cerebrospinal fluid. Lately also we have been developing a technique where we actually can go through the ear, and that sounds a bit odd. It's used in otosurgery when you want to remove, for instance, a tumor that grows centrally in the brain. Earlier we had to go through the mouth or through the neck, and what happened is that you destroy a lot of healthy tissue, nerve endings, cranial nerves who are damaged and so on. But then they came up with the idea, well, let's try to go through the ear. And of course, Then you have a surgical team around this that do it, and it takes about 15 hours, the whole procedure. But you open up the ear, you put the outer ear as a flap on the back of your head with plastic surgeon, and then you drill out a piece of the bone. It looks like kind of a device you use when you put electric plugs in the wall, you know. And then you come with a small dentist drill, and you go millimeter by millimeter into the inner ear, and then you enter into the brain and can remove the tissue then. And we have developed a small sampling probe, so it's a small thin, like a needle basically, but it's coated on the surface with selected molecules that can actually draw and enrich certain markers then of interest from the tumors. So we actually do that through this entrance into the ear. And after the surgery is done, the patient will be have plastic surgery again, put everything back, and then you can restore the hearing by a cochlear implant. So they actually don't lose the hearing on that side either. So that's a fantastic change compared to what was done earlier. Then, it's very hard to see here. Biopsy materials. So this is a thin glass slide with some materials on. It's of course used. When we have the option to get biopsies, it's fantastic. So brain biopsies very rarely, and from healthy material it's almost impossible to get. But we do it with peripheral organs. So muscle biopsies is very much used when it comes to neuro-inflammatory reaction markers for disease, or where we have a loss of innervation in the muscle. So for instance in amyotrophic lateral sclerosis, which is called Lou Gehrig's disease here, then we take biopsy from muscle. But then blood is of course something that is super available, so blood samples, and that's what we are now looking into when it comes to specific patient groups where it's hard to get any of the other tissues or samples. A simple blood test, maybe a single droplet of blood is the target. And then we use a technique called dry blood spot selection. So we take a capillary blood or venous blood and let one droplet dry down on the surface on a paper, and then we use that paper for analysis later on. So that makes it available also for patients that cannot go to the clinic, for instance, or are somewhere else in the world. So I just told we have a collaboration with South Africa now, and some parts of South Africa don't have clinics for sampling, and then they can get a sampling kit sent to them, and they do the sampling at their place and send the samples back to us for dissection. So cerebrospinal fluid then. Yeah, this slide, you see a skull here. You see a cheek here, and here you see the blue. It's not blue in real, it's actually clear liquid, but the blue liquid here is the cerebrospinal fluid. So it's produced centrally in the nervous system, and we produce about 500 to 600 milliliters per day of this liquid. So where does it go? Can anybody tell?
Spine?
Yes, it flows down around the brain and then along the spine, and then
I think I might have lost it.
yes. Sweat. No, it actually is resorbed. So we are resorbing this liquid all the time. So it's turned around several times every day. So this liquid cleans up the brain. It acts as a mechanical protection inside the skull. Otherwise the brain would just wobble around inside the skull. And it also keeps clean of waste products and metabolites that are released from the brain. So if we can get a sample from this by lumbar-puncture or going into the ventricles in the neck, we can actually monitor the chemistry of the brain with a very nice way of doing it. But as I said in the beginning, yes?
What is it? The kind of liquid? Can you produce it by mechanic and put it in, but just the body can produce it?
Yeah, the body produces itself. So there's a gland here called choroidus plexus.
What's in it?
That's proteins, yeah, mostly water, yeah, but it's proteins, small metabolites, and waste products from the metabolism or turnover of cells. So, and if you have a disease, we have a specific set of markers. So for Alzheimer's patients, for instance, we can monitor a certain set of markers that tells this patient has Alzheimer's disease and at what stage this Alzheimer's disease is. Is it a other type of dementia like vascular dementia or frontal lobe dementia? We can tell by looking at these different patterns.
But you can't put in, I think you put in with antibiotic or something like that. You took it out from that. And
we draw it out to measure things. We can administer drugs this way also to get it into the ventricle by a probe or actually infusing it in the neck. But the thing is that the flow of this liquid goes from the central part out.
Oh, yeah.
So it's hard to get things into the brain that way. You have to you can use vesicles that go through membranes and so on. Then you can get it into the brain.
So you said you could diagnose deceased people's, what stage Alzheimer? Can you do that with living people too?
Absolutely, these are living.
Yeah, because I've been told that you can only diagnose by a doctor, based on the family's observations and this and that and the other. But
that's where you start because.
Gotcha.
Yeah, but I mean, in principle, if we could screen everyone, we would be able to tell to a certain degree early onset. So there are other ways of, you look at the clinical picture, you have a set of mini-mental tests, you ask questions, you ask your relatives and so on, and look for behavioral changes and things like that. So this is just the chemical part of the diagnosis, and it's all a teamwork really around all patients.
Gotcha.
But it's a pity it's so hard to see here. I wanted to show you something which I think is pretty cool. I'm a bit biased though. Let's see if I can get this started here anyway. So actually what you were supposed to see, maybe you can see it actually. Can you see that this brain is actually moving, right? What do you think that is? It's pumping.
Falling off a horse.
Yes, partly. I have to say this is a bit exaggerated. So this is actually how it looks like, and you can't. If you look carefully, you can see that it's actually moving over here. But they have exaggerated this MRI picture. There's a functional MRI picture of the brain on the patient. Actually, it's the beat of the heart, and the pulse actually now has been found that it actually pumps this liquid around, and it also connects to this lymphatic system, as I said, so cleaning up the brain. So this pumping motion based on the blood pressure will actually help to get the flow in the right direction down to the lower spinal region, and the resorption goes on the way. If we don't resorb, we get hydrocephalus, you know what that is. In children, the skull will grow and you will have overpressure. In an adult, you will get, we call it normal pressure hydrocephalus sometimes, and that's also not very high pressure inside the central region of the brain, but the ventricles can be a bit enlarged, and the patient will react with fatigue, so they will be very tired, have problem with motion, mobility and so on, and sleep 23 hours a day. If you go in and reduce the pressure, evacuate some of the liquid and put a small catheter here, so you actually help releasing the pressure from the central nervous system, they wake up and they go back to total normal life. So it's fantastic to see those patients really going from totally immobilized to full motion and full activity again. So if you can target those in time, you will have a good possibility to save these patients. So what we use as a tool for doing the molecular diagnosis on patients is called mass spectrometry, and I won't bore you too much with that technique, but it's a technique that can monitor and measure thousands of molecules at the same time in a complex mixture, and it can tell us exactly what is the name of the molecule, how much of the molecule do we have, what is the structure of the molecule, is it modified in any way. So we can do that, and we can do it for thousands and thousands of molecules at the same time. So that's the beauty with the technique. A problem with the technique is that it's super sensitive. So if there is any contamination or anything, someone is not behaving well with the sample, the mass spectrometer will tell us, and then we have to maybe do a new sampling and set up a new test and so on. But it's a fantastic tool, and as you see here maybe in the title, we call what we are doing proteomics or metabolomics, but it's basically protein analysis or metabolite analysis, but in a large scale. So that's the term omics comes from that. So I just wanted to dive a bit into the metabolomic part, because I think that sort of relates to what we are doing here a bit. The metabolism in the body is central and essential for us, and it also affects our phenotype. So who are we basically, based on what we present as our metabolites. And it's very hard to see here on the screen, but things that will affect our metabolites or metabolomics is our genes, what we get from our parents, and also the epigenetics, which is modifications we do to our genes during our lifetime. But there are other things, of course, that affect our metabolism, like food intake, what do we eat, what kind of activity do we have, do we take any drugs, do we have an illness, etc., etc. And lifestyle down here, for instance. So that gives us this complex picture of the chemistry in the body, and also tells us what is actually going on if you have a disease, or if your lifestyle is not the best maybe, and you have something you have to modify in your lifestyle to avoid getting a disease. So when it comes to nutrition, I found this slide, which I think is pretty interesting. Someone had a bit too much time, I think. They had correlated the numbers of Nobel Prizes and the consumption of chocolate. And you can see there is a very nice correlation here, that Switzerland and Sweden, we are on the top, we have most Nobel Prizes and consume most chocolate. US is doing pretty poor. China is worse. Ireland is up here somewhere, right? Where is it? There it is, Ireland, Germany. So you are on the rise here, but still you have to fight this a bit, I think, to get up in our class.
I have to take a picture because I like chocolate.
Yes. And chocolate. Why is chocolate interesting in this case? What does chocolate give us?
Sugar.
Antioxidants.
Sugar, antioxidants, fat, cholesterol, things like that. We hear a lot about cholesterol, how damaging that could be, but remember 50% of the brain is actually lipids, so we need lipids. Of course they need to be in the right place. We shouldn't have them here or too much here, but very important to have enough of it. And if you have starvation in children or young adults, that can be devastating, and that will also affect you for the rest of your life. But of course, it's always, as we say in Sweden, it should be lagom, which means just enough, not too much, not too little. It should be lagom. So if you have the right lipids, that's perfect. There's something else in chocolate, which is actually making a lot of us happy with eating chocolate. And what is that?
Cocoa.
Yeah, but what's in cocoa?
Caffeine.
Coffee? No, not cocaine. No. Phenylethylamine. That's a molecule that looks very much like amphetamine. The chemical structure is very, very similar to amphetamine. And that's our lucky molecule. So that's quite a lot of it in chocolate. Problem is that when we get it per oral, and intake from our transportation from the gut to the brain of this molecule is not super efficient. So it's actually not the reason why we eat chocolate. But it's exactly what you said. Combination of fat and sugar. So those two in a right combination makes the brain happy. And then the brain itself starts to produce phenylethylamine, our lucky molecule basically. So if you look at the bad parts a bit, stress, stress is affecting us in a bad way. Quite a few of us need a bit of stress to keep us activated, but we should not call that stress. They sometimes say positive stress. I think we should call it stimulation instead, because that's more direct and true way of describing it. But stress is terrible in many ways. It affects the brain, the cognitive behavior of us, and it affects our ability to learn and to live our lives basically, and to grasp things around us. It affects our cardiovascular system, so heart, problems. It affects the gastrointestinal system, so stomach and intestines will not work well. And you know, if you're stressed and nervous, you know your bad stomach is very common. You have pain in your stomach, or you get well, you can both have constipation or diarrhea or whatever, and you feel not well. Then, which it says here, pancreas. You know, the pancreas is producing the insulin and controlling our blood sugar levels, and if we are stressed, we also affect the pancreas and also cause severe stress, also cause inflammation in pancreas, and that can in the long term lead to diabetes, for instance, so also affecting us severely. Yeah, we talked about that, and our steroid hormone regulation is also very much affected by stress. Now we talk a bit more about that later also. So what we are involved in right now, not only looking at the diseased patients with different neurodegenerative diseases, we also try to figure out why is there regions of the world where actually neurodegeneration is less, because maybe we can learn something from those places. And there's something called the blue zones, and here are a few of those. There are actually more than these 5 here. You see the blue dots here maybe on the. So there is an island outside of Japan, for instance. There are some islands in the Mediterranean. There is somewhere in Central America. And there is actually up in Sweden a region also where people grow very old and live happy lives in some way. That's called the blue zones. And one of these studies going on now to understand what is happening here. Why are they happy? And if you make small short list of this, diet has been proven to be very important. And as we say in Sweden, lagom. So not too much of anything, not too little of anything. But of course, good food, good fresh food, not too much processed food and so on. And I think that is well aligned with what Christian and Macrell is doing with their horse feed now, trying to avoid things that are not good for our horses, and also get that diet to help the patients or horses patients to actually become better and improve with reduced inflammation, etc., etc. Exercise, very important, and this is what we do all the time if you have a horse, definitely. But it's important not to think that you have to do this massive exercises, running marathons or whatever, because that's actually not so healthy. Some people do it anyway and like it so much, so I guess that's sort of healthy in that sense. But what we are thinking of when it comes to exercise is this normal daily low-grade exercises, but something you do all day or every day.
Like riding.
Riding, definitely. Very good example. Because you need that. You need to get circulation going. You need to help metabolism going and take care of the food that your food intake. So there's a balance between intake and output, basically. Social behavior, super important. And that's the 2 last points here, I think, has been sort of neglected in some way, because social behavior, meeting, interacting with other people is super important. So I think these kind of events, for instance, is a good example. You learn to meet new people, you have discussions, you socialize with each other, you socialize with the horses. And now over the years we have set up methods for monitoring things. So we can take one single droplet of blood and we can monitor. Today we have methods, targeted methods for about 800 different molecules that we can do, and they are lifestyle related. So we can actually monitor the health state of an individual by just looking at the content of a single droplet of blood. And I think that's something also attractive, because everyone probably would be happy to give away one single droplet of blood or a few single droplets to be monitored for different things. And you can't read the list here, I'm sorry for that, but I just give you one example. Here, down here we have steroids and steroid hormones, and they are essential to what happens in life, basically. It controls the metabolism, controls our fertility, for instance, when it comes to having babies. It controls heart, brain, all the endocrine organs in the body controlled by these molecules. And they are small organic molecules. They all look basically the same, but we monitor today in our method about 40 different steroid hormones. And the most common one, I think you all know them, estrogen, testosterone, cholesterol, cortisol. But there are, as I said, about 40 of these markers. It's estrogens, as you know, is what we call generalized a female hormone, but it's actually very much a hormone that exists in all of us. Females generally have higher levels of estrogens, while males have lower levels. Females have low levels of testosterone, while males normally have higher levels. But that's sort of generalized. Hormone regulation is super complex, and that's what we now realize. Early on, we could only monitor one single molecule at a time. Now, when we can monitor a whole pattern, we understand how this complex network of metabolism actually is involved. And things that we haven't been able to see before, when it comes to, for instance, testosterone regulation in women or children. They have extremely low levels sometimes. We have not been able to monitor those before. And now with our technique, we can go in and actually look at what is happening. So we can explain, for instance, some metabolic disorders by doing this monitoring. We can figure out what is going on when you have infertility problems. We can monitor cancer markers based on these steroids. So certain breast cancer types, for instance, have a down or up regulation of certain steroids in the pattern. We can tell from that pattern what kind of breast tumor is growing and what is the possibility for treatment, what's the best treatment, who will have side effects of certain treatments and who will benefit from certain treatments. We can do patient selections on these. Hormone regulation is pretty complex, and I found this picture here. It's Swedish, but I translated it. It's a guy sitting here looking at a model in a croquis course, and he says here, it wasn't until I took this evening class in croquis I understood how complex women are, right? He's doing a pretty crappy job making a picture of her, right? So, female hormone regulation, that's, I think, between males at least, we think, oh, they're so complex and they're so difficult to understand sometimes. This is the equation, actually, so you can put that in a mathematical formula. This explains the hormone regulation in women.
There is an answer.
Yes, there is an answer. And it's not so difficult, actually, if you look at it. It's pretty straightforward. It's cycles. And you can actually express that as an equation. So how does a male version of the hormone equation look like? The flatliner. No. This is how difficult the male regulation of hormones are. So this is actually 7 equations needed to put together to actually create this pattern. So we are pretty complex, actually.
More complex than the other one.
Yeah.
Better made.
So that's it.
Researcher was made.
Yeah. So this is things we have to figure out, right? What's going on and how are we doing when it comes to different hormone regulations. So that's why it's so important to be able to monitor more than one thing at a time.
What about castrated males?
Yeah, actually, it will shift.
Okay, how is some...
Yeah, actually, that's interesting. They've done some studies on changes of steroid hormone regulations. They don't differ that much, actually. So the only thing they found so far is that certain receptors are located a bit different and a bit different density or amount of receptors. That's the receivers of the signals in the cells. They are a bit different, but not so much. So it's more a behavioral thing in that sense. And quality of life then is where we come down, what it all boils down to. So make sure that you get a good quality of life. That sounds also easy. And this picture is from Japan. I saw this little lady walking across a rice field here, and she looked like she was like 150 or something, or bent over. But she looked, and I met her, she looked so happy with life, basically, content with her life. And of course, not all of us will go and be 120 years of age, and maybe not that's maybe not the goal either. To have a good quality of life is important. And that leads us into horses. And here's a picture of our 2 daughters with our 2 horses. Prinsen and Morgana, and Molly and Nanna, our 2 daughters. And I think some of you at least know a bit about what equestrian therapy is and how it can be used. So equine assisted therapy can be, for instance, used, and now it's been documented to be used in some neuromuscular diseases, in cerebral palsy, multiple sclerosis, MSA, which is also a muscle neurodegenerative disease, to keep up training a bit, help with balance, and also stimulate the muscle. But it also been used as a diagnostic tool. So if you have a muscle disorder and a neuromuscular innervation problem, you can actually put the patient onto a horse, and then, at least I haven't seen the data on it, but they describe that you can actually tell based on how the horse moves, it sort of reflects the rigidity or instability in the patient. So you can actually use the horse as a diagnostic tool. I'm not sure if that's really been proven, but it's a nice idea to think about. So we'll see about that. But then there are other things here, and that's more soft science, I have to say, demonstrate on this plot here. So for instance, self-esteem, behavioral, social interaction, help for recovery. When it comes to neuropsychiatric diseases, now we have 2 neuropsychologists in the audience, so you can help me put something into this probably. But working with horses or living around horses and with horse people, if they don't drive you crazy, is helping us in many different contexts, and I won't read off all of these things. But what happens with us when we go out and start working with a horse, also if you like horses, is that your brain starts to work in a very nice way. This is actually my brain. It's an MRI picture I got. I was part of a study as a control I have to put in.
Have you make it big on the screen? No.
No, it's you see, it's very dense here actually. It just fills up the whole skull, so it looks nice. But now it's hard to see. Sorry for that. But one region that lights up when we start working with the horse is this green part here, which is below the ventricles, the hole where we have the central, the cerebrospinal fluid produced. So that region is lighting up if you have a monitoring of the brain in a brain imager, like MRI or PET image, positron emission tomography picture. And what happens here is, oh, my time is out. What region do you think that is of the brain? You don't need to know the name of it, but what's the part of the brain? Sorry? Hypothalamus, yeah, not too bad. It's but what is the region? What is it doing for us?
Creating that fluid.
Yeah, it's below that. What happens when you go out and start working with something you like?
Motor skills.
Sorry?
Motor skills.
Motor skills?
Serotonin creates the hormone that's in charge.
Tillfredsställelse, reward, right? So this is the reward system. This is one part of the old part of the brain called the reptile brain, right? So the reward system is there, and when we trigger the reward system, it lights up, so it goes active. And you can tell actually when you do that with different stimuli that this region is very important for us to enjoy what we're doing. And there is a set of molecules that are very important to us that is produced in that region. You know, probably dopamine. Dopamine is one of the molecules that are making us feel good and also stimulates us to do more of it. So it's sort of a dangerous molecule in some way because it can trigger us into bad behavior also, like drinking, smoking, using substances that we are not supposed to use and so on. But also natural behavior and induced reward is very important for us, and that makes us happy, content with ourselves, and we enjoy it, and we go further and further into using this region of the brain. Another part of the brain that actually probably is going to be activated in most of us is this one. It's a frontal part of the brain. What do you think is happening in that part of the brain? Reasoning, yeah. Judgment. So that's where the judgment area is. And what happens when you look at it, people who go into, let's say, risk behavior in some way to trigger the reward system, they also reduce their judgment a bit or break on the judgment. So what happens is we take a bit bigger risks, and we also are prepared to actually, for instance, do economically maybe not totally reasonable things. I think all of us know that. So important though is that if we manage to lower our control, our frontal lobes a bit to actually help us to induce the reward in the reward system, that will trigger certain molecules. And we have been discussing some of these molecules. Oxytocin is one molecule that some of you have heard of. It's a bigger molecule, it's a peptide, so it's small amino acids put together on a chain. And this molecule is released when we feel comfortable, when we have reduced our judgment a bit, take some risk, get the reward feedback, and then actually get production of oxytocin. Oxytocin is also released when you breastfeed a baby and the mother, when you touch someone. So if you get touched by someone or someone touches you, if you pet a horse, for instance, you start producing oxytocin and the horse starts to produce oxytocin. So it's very important for us in the way of connecting to each other. So relationship is basically triggered by oxytocin. Important for us. Then there's a red region here where it's very hard to see. It's a bit smaller region just next to the reward system. But there is one other region here. And what is that region? I think part of us will also activate that region. Some of us have already done it, I think. Fear, yes. Good. So there is a region of fear. And that one we have to fight a bit if we are going to explore new things. So we have to be careful with it because it's important. It's a safety system for us, but it's also something we have to fight and push. So as when Daniel says, well, you go as fast as you can and then you just do a bit more, then you actually suppress this fear system and the judgment system, and that in combination leads to a bit of a risk behavior, but makes you happy in the end. And I was thinking, should I have this region? The last region I will go through today is the yellow region up here. What do you think that yellow region is? And I hope definitely that none of us will trigger that today. What's that? And you see, it's a pretty big region also.
Sadness.
Sorry?
Sadness.
Sadness, no. Yeah, that could, but that's not that region actually. Hopefully no sadness either.
Hopefully.
Yeah. Pain. So pain is controlled and registered in this region of the brain here. And it lights up if you have pain. Interesting enough, if you do open surgery of the skull and you take a needle and you push it into the brain, it doesn't hurt because the brain in itself doesn't have any pain receptors. So that region, called the periaqueductal gray matter, it actually records all the pain sensation we have in the body. But in the central brain, we don't have pain receptors. When we have headache, it doesn't come from the pain receptors in the brain. It comes from microvesical contractions, for instance. You have no oxygen in certain regions of the brain. And we record that as a pain signal, but it's not pain in itself, basically.
Why can't you go in and make that a little bit smaller and happiness a little bit bigger?
But we do that. We try to do it, and we do it by our activities. So when you are stimulating your reward system, you will, and you get something that is very beneficial for your health. Self-esteem, super important. To get self-esteem and to always try to appreciate what you do yourself, to try to make yourself feel a bit appreciative to yourself. We are so good on putting ourselves down, right? So try to do better on that side and have a good feeling about yourself. It's important. It can be easy to say, hard to get sometimes. But then I think these kind of training events, for instance, I mean, if you know, okay, I picked up a bit, I was maybe doing a bit better this time. Daniel looked a bit more happy now this time and so on. Then, okay, you can feel proud of yourself. I think that's important. And take all the small moments you get of that and boost yourself. Then there will be things happening that is not so good, but you should try to forget those as fast as possible, basically.
I think Americans are so much better on this to encourage themselves and to encourage each other than we are in Sweden.
Absolutely.
We have
a bit of, we call it Jante law. You are familiar with that? The Jante law is like you should not boost yourself too much. I mean, it's like it's a bit. Exactly.
Yeah, already as a kid, it's kind of like if someone was trying to brag about something, you're proud of something. The other kids would say like, "Don't think you are something."
Yeah, exactly. Just.
And that's the word you can do exactly like that. And you start off, and then like you would be happy with something accomplishment. You're a good test, and then say, "Don't think you are something."
And that's not good. It's not healthy.
No.
It's not.
It should be a balance though, because if you know you're good, you don't need to brag too much about it, right? So it's more like what you feel yourself is important. So that's important.
Boosting systems that you are triggering. While, as we said in the beginning, if we take out any stimuli to a certain region, it will waste. So if you would lose that, then unfortunately, it's like this. Like we lose after 23 years of age, if we generalize, brain goes down. It goes slowly degrades and you lose brain tissue. But we compensate that with our knowledge instead, right? Experience.
Wisdom.
Wisdom. Exactly, wisdom is important. So that's how we deal with the situation. Another mathematical slide here, which is very hard to see, I know that. This is some guys who have made a calculation and equations for how we behave in a relationship. And it could be a relationship with your partner, but it could also be a relationship with your hobby. So let's assume this is the hobby then. But they made calculations with steroid hormones. We can actually monitor where you are in a relationship. So if you are living like this with a rather flat structure of your relationship, if you are on the peak of a relationship, or if you're like bimodal like this, up and down, or like this, which seems
so that's jumping in the bottom and flat work on the top.
So that's actually possible to monitor with the techniques today, which is pretty cool actually. A bit scary also. So time is flying when we have some jumping to do here, so I don't want to take your time too much. But what we are looking at now is possibilities to monitor this and apply that to horses also. So we have set up, for humans, we have set up a number of test methods basically. We can ask for certain things based on single droplet of blood. So if you want to monitor,
so we can ask for certain things based on single droplets of blood. So if you want to monitor your stress hormone levels or metabolic functions of thyroid glands or whatever we do, we have vitamins and minerals and so on. Pre-diabetes tests. So what we are now targeting is other species. So we're cat kit, dog kit, horse kit maybe. So actually go and monitor things. And then sampling will be an issue in some way. We have to figure out what is the best way of sampling. Single droplets of blood can be simply drawn from cats and dogs and horses. So that would be one way. You can do lumbar puncture on a horse. It's not trivial, nothing you do on a regular basis. You can do it when you sedate the horse, put it on the side, and you go in the lower region of the back. You can also have them sedated standing up, and you can do it in the neck or in towards the tail basically. But it's pretty tricky to draw cerebrospinal fluid from a horse. So I think that's not the way we are going to monitor things in most animals. If we had the pig here, I could show you how to do. The pig, we have done pigs quite a lot, and rats and mouses, of course. Dogs we have done, and cats. And we have horses done right now, basically, on just to screen for what markers we get in cerebrospinal fluid. But for general use, it would be a blood-based test for sure. Or it could be fecal samples, urine tests. Could be also a possible way. So I'll finish up with this. This is the love test that we're going to administer also. So where are you in your relationship? You buy kit, you get 2 tests, and then you test each other and then send them in for analysis, and we'll see what happens. I don't know. Yeah, that could be. Probably this is not going to be hitting the market yet, but we'll see. Maybe we can use it like horse owner and horse, and then see if there is a match or not. That would be more ethical. So, thank you very much for your attention. And I'm happy to answer some questions if you have.
Do we get continuing ed credit?
Sorry, once again?
Do we get continuing medical education credit?
Yes, absolutely. We'll sign credits for you. So since we have the opportunity to ask our neuropsychologists also, what's your, I know you both work with children. What's your opinion about using equine therapy in children for behavior changes? In autism or Asperger or...
Well, most of the research is anecdotal, so there's not been really well-controlled studies. But it's still kind of in the early stage, so I think there's some promise for that. The VA has done some funding for working with veterans and post-traumatic stress. And there's some, considerable side effects with those individuals, especially for PTSD. But a lot of stuff again is anecdotal, so it lacks kind of statistical power in that regard.
Yeah. So what we are doing in...
It has been used for a long time, not that structured though, not in therapy, but it's used for cerebral palsy.
Absolutely.
Muscular
balance and...
exactly.
Yeah, and again, like on an equal one basis, another barn, there was this girl who was wheelchair bound. She had pretty severe cerebral palsy, and over the course of several years was riding and doing, riding independently. It could do the rising trot, and it was pretty impressive. And she was also ambulatory, so without any kind of support. So that was pretty impressive.
It takes some studies to realize that if you can't move on your own, suddenly you're able to move on your own. It doesn't take a study to see that is beneficial.
Exactly.
Absolutely. Yeah. And the stimuli, that is important, right?
But
the
then
positive
the
stimuli.
outcome measures for things like, autism and Asperger's, and those kinds of neuropsychiatric disorders are a little harder to define.
Yeah.
So, but
Yeah.
I want to say one thing before we turn off.
Yes.
So, just with the brain activity that we know that's going on with horses, one of our big wishes, and that luckily we have all of you guys to help us, is to find ways to bring non-equestrians into contact with horses, whether it's riding or therapy for someone with a diagnosis or not a diagnosis, and just using horses for regular stress management, because we know how beneficial it is for all of us. So all of this is the things that we're doing with Requilife are not just for equestrians, but also for non-horse people that haven't had the privilege of interaction, because we know that it works. All of us, we do this not because it's cheap. We do it because we get a whole lot out of it, right?
Yes.
Perfect. Thank you so much, Jonas. That was amazing.
Thank you for being here.